Hello my friends, now we are going to discuss about the hydrogen production from the biological process. So, in our past 4 lectures we have discussed about the productions of the hydrogen by different process like electrolysis process, by fossil fuels or by some other means. In this particular case we are going to discuss about the hydrogen production from the biological process itself. So, in introductions we can say that hydrogen is the fuel of the future, because now so many companies like Toyota or maybe in some kind of space shuttles, basically people or maybe scientists are trying to use the hydrogen gas as a fuel. So, this can be used in a future fuel, because as the petroleum products or maybe the fossil fuels are going to be finished within some couple of years. So, basically then in that particular case we have to totally depend upon the renewable energy, and this kind of renewable energy, the hydrogen productions and used hydrogen as a fuel will be the most efficient one. So, basically hydrogen is the fuel of the future, as I told already, mainly due to its high conversion efficiency, recyclability and non-polluting nature. About half of all the hydrogen currently produced is obtained from thermocatalytic and the gasification process using the natural gas as a starting materials. But in this case also there is the same problem persist, that we are using the natural gas. So, natural gas also the limited one. So, maybe after certain time we have to work on something like that which can be directly coming from the nature as a waste product like sunlight or maybe some kind of energy or maybe some kind of electrolysis of the hydrogen, so electrolysis of the water, by which we can produce the hydrogen gas. So, large quantum of waste generated from diverse source, especially food industry and agricultural practices, seems to be a viable feedstock for biological hydrogen production. Biological hydrogen production process are found to be more environment friendly and less energy intensive as compared to thermochemical and electrochemical process. So, basically what we are talking about? We are talking about some kind of bacteria, some kind of virus, some kind of algae, those basically are generating the hydrogen gas. If we are able to capture that hydrogen gas and store it, so automatically that generation, hydrogen generation is the waste one, directly it is going into the environment. So, we are not able to collect it. So, now the scientists are thinking to collect those hydrogen gas as a future fuel. So, basically we are having certain kind of bioreactors or maybe we are having that fuel cells. So, basically by sunlight, that photo bioreactor, it is producing the hydrogen gas, or maybe the breaking of the water into the hydrogen and the oxygen, we can collect the hydrogen gas. So, they are producing certain kind of hydrogen gas over there, and another beauty of this particular technology is that simultaneously they are absorbing some kind of toxic gases like carbon dioxide, carbon monoxide into the systems. So, one way they are reducing the pollutions from the environment, as well as they are giving the hydrogen gas. So, what we are trying to store in some for the future applications. So, now if we talk about the history, so you can see in the year of 1939 Han Gaffron discovered that algae can switch between producing oxygen and hydrogen. Then in the later in the year of 1997, Professor Anastasios Malis discovered that deprivation of sulfur will cause the algae to switch from producing hydrogen. He found that enzyme hydrogenases responsible for these particular reactions. Then we have come to in the year of 2006, researchers at University of Bielefeld genetically changed the single cell Chlamydomonas reinhardtii in such a way that it produces a large amount of hydrogen. So, this is a one kind of biological product. In the 2007, Professor Anastasios Malis, studying solar to chemical energy conversion efficiency in tax X mutants of Chlamydomonas reinhardtii, achieved as 15% efficiency. So, now you can understand that how the scientists are working to produce the hydrogen from the biological product. So, biological hydrogen production process, in short basically we are calling it as a BHP. So, BHP from renewable source like biomass, water and organic wastes either biologically or photobiologically is called the biohydrogen. Biological production of hydrogen as a byproduct of microorganisms metabolism is an exciting new area of technology development that offers the potential production of usable hydrogen from a variety of renewable sources. BHP at ambient physiological conditions is the most obvious and viable approach over energy intensive conventional chemical or maybe the electrochemical processes. A successful biological conversion of biomass to hydrogen depends strongly on the processing of raw materials to produce feedstock which can be fermented by the microorganisms. BHP can be realized by anaerobic and photosynthetic microorganisms using carbohydrate rich and non-toxic raw materials. Now, we were not thinking that we can produce or maybe we can be able to produce the hydrogen from these particular bioorganisms or maybe from these particular microorganisms. So, now people are tending their research towards this field. So, basically they are trying to make certain kind of microorganisms which can produce the hydrogen gas, and that hydrogen gas in for future applications we can store. Now, what is the classifications of biological hydrogen productions? So, basically it has been divided into 5 parts. First one is called the bio photolysis, then second one is called the fermentations, third one is called the biological water gas shift reactions, fourth one is the microbial electrolysis cell, and the fifth one is called the major enzymes. Then if we go into little bit deeper of the biophotolysis, it has also divided into 2 parts. So, direct biophotolysis and another one is called the indirect biophotolysis. If we talk about the fermentations, it has also been divided into 2 parts, one is called the photofermentations, another one is called the dark fermentations. So, this is the whole classifications of the biological hydrogen production. Now, we are going to discuss one by one. So, first is called the biophotolysis. So, first we are going to discuss about the direct biophotolysis, then second one we are going to discuss about the indirect one. So, biological hydrogen can be generated from plants by biophotolysis of water using the microalgae, green algae and the cyanobacteria, fermentation of organic compounds and photodecomposition of organic compounds by the photosynthetic bacteria itself. So, photosynthetic production of hydrogen from water is a biological process that can convert sunlight into useful stored chemical energy by the following general reactions. Simple, the water is breaking in the presence of sunlight or maybe the light energy into 2 hydrogen and the oxygen, that means into the hydrogen gas and oxygen gas. Simple splitting is taking care by this particular plant or maybe by the plant itself in the presence of sunlight. So, the light energy is absorbed by the pigments at photosystem 1 PS1 and photosystem 2 PS2 or both. So, in this case this is the PS1 and PS2 both. So, which raises the energy level of electrons from water oxidation when they are transferred from PS1 via PS2 to ferredoxin FD. The hydrogenases accepts the electrons from FD to produce the hydrogen itself. So, in this particular case it is PS1 and PS2 systems, directly solar energy is coming and then we are using these ferredoxins over there or maybe it is already present over there. By this hydrogenesis is taking place by which we are getting the hydrogen gas, and in this particular case the oxygen is coming throughout it. The concept of direct biophotolysis envisions light driven simultaneous oxygen evaluations on the oxidizing site of PS2 and hydrogen productions on the reducing site of PS1 with a maximum H2O2 ratio of 2:1. Such reactions with green algae could serve to provide a clean, renewable and economically viable hydrogen fuel. Since hydrogenesis is sensitive to oxygen, green algae C. Reinhardt T is used to maintain the oxygen content at a low level under 0.1% so that hydrogen production can be sustained for a longer time. Now, we are going to discuss about the indirect bio photolysis. So, cyanobacteria can also synthesize and evolve hydrogen through photosynthesis process via the following process, like 12H2O plus 6CO2, simple it is preparing the glucose C6H12O6 plus oxygen. Then this C6H12O6 glucose additions with the water, 12 molecule of water, then it at light energy or maybe the in presence of sunlight, it is producing the hydrogen gas and the carbon monoxide gas. Indirect biophotolysis therefore consists of two stages in series. One is called the photosynthesis for carbohydrate accumulations, this is the number 1, and number 2 is the dark fermentations of the carbon reserve for the hydrogen production, that is the number 2. So, basically indirect biophotolysis process involves separations of the hydrogen and oxygen evaluation reactions into separate stages coupled through the carbon dioxide evaluation. The cells takes up carbon dioxide first to produce the cellular substances which are subsequently used for hydrogen production. So, this is about the indirect biophotolysis process. Now, we are going to discuss about the second one, that is called the fermentations, that also divided into two parts. One is called the photofermentations, another one is called the dark fermentations. So, in photofermentations, basically the photosynthetic bacteria evolve molecular hydrogen catalyzed by nitrogenase under nitrogen deficient conditions using light energy and reduce the compounds itself. So, the overall reaction of hydrogen production can be given as same, it is like glucose, then we are adding the water, and in the presence of sunlight or maybe the light energy, it is producing the hydrogen gas with carbon dioxide gas. Many studies have demonstrated that photofermentation by photosynthetic bacteria can convert small molecular fatty acids into hydrogen and carbon dioxide with high efficiency. So, basically we can do by these methods, it is called the sugar cane. Suppose we are taking and then we are doing the milling. So, milling the extract, basically it is known as the bagasse or maybe the bagasse fiber. Then after milling we are doing the photofermentations. So, in one case we are using the anaerobic digester and we are able to produce the methane gas. In other case we are using the pressure swing adsorption techniques by which we are able to produce the hydrogen gas. Now, the next one is called the dark fermentations. So, basically the dark fermentations mainly differs from photofermentations in which it works without the presence of light. So, that means we can do it into the lab or maybe inside the room. So, DF employs diverse group of facultative and anaerobic bacteria such as E. coli, E. cloacae and Clostridium sp for the efficient conversion of wide range of organic substrates. DF technology has the simpler reactor design and less energy requirement when compared to other hydrogen production technology. So, the most dominant hydrogen producing route for the DF can be achieved with the acetate mediated fermentive pathways with the generation of 4 moles of molecular hydrogen with 1 mole of hexose as shown in equation. So, in this particular case what we are trying to do? So, we are basically trying to do the fermentative substrates such as biomass or maybe the agricultural products or maybe other organic wastes we are collecting, then we are doing certain kind of pre-treatment over there, pre-treatment in terms of some kind of cleaning, some kind of pre-heat treatment kind of things, then we are doing the fermentations and through fermentations simple we are getting the two gas, that is the hydrogen and the carbon dioxide, and simple we are separating the hydrogen gas and the carbon dioxide gas by the gas separation process by which we are able to produce the hydrogen gas. So, this is the overall thing about the dark fermentations. Now, we are going to the third part which is called the biological water gas shift reactions. So, hydrogen is produced via water gas shift reactions by the photoheterotrophic bacteria. So, while carbon monoxide is oxidized to carbon dioxide in the presence of anaerobic bacteria, hydrogen is released from the water gas shift reactions shown in this particular part. So, carbon monoxide in the gaseous state, H2O into the liquid form, it is forming the carbon dioxide as a gas and hydrogen as a gas. So, in this particular case organisms growing at the expense of this process are the gram negative bacteria such as Rhodospirillum rubrum and Rubrivax gelatinosus and the gram positive bacteria such as Carboxydothermus hydrogenoformans. So, basically these all are the names of different bacteria. So, basically bacteria is divided into two parts, one is called the gram positive and the gram negative bacteria. So, basically in this case the help of this kind of bacteria we are trying to develop the hydrogen gas from the biological water gas shift reactions. Next, the fourth one is called the microbial electrolysis cell. So, in short basically we are calling it as a MEC. So, MEC is a novel bioelectrochemical tool for hydrogen productions that employs domestic and industrial wastes as fuel source and used electrochemically active bacteria EAB as a biocatalyst with the presence of electric current. So, simple in this particular case what is there? We are having two electrodes, one is known as the anode, another one is known as the cathode. In between that we are using the separator membrane. So, basically we are using some kind of polymeric membrane over there, and one side we are giving the electrochemically active bacteria. So, this is basically the electrochemically active bacteria and it is attaching with the anode materials, and then after that what is happening? MEC consists of anode, cathode, membrane, electrochemically active bacteria as an electric power supply. So, this is the external road that by which we are giving the potential difference in between the two electrode over there. Now, hydrogen production efficiencies of MECs generally 80 to 100% are significantly higher than that of the DF process, that is 33% and the water electrolysis of 65%. Now, you can understand that what is the efficiency we are achieving by this microbial electrolysis cell. So, basically what is happening, we are able to achieve almost 80 to 100% efficiency for the hydrogen production. Next is called the major enzymes. There are three fundamentally different hydrogen producing and metabolizing enzymes found in algae and the cyanobacteria. First one is called the reversible or classical hydrogenesis, second one is called the membrane bound uptake hydrogenesis, and the third one is called the nitrogenase enzymes. So, first we are going to discuss about the reversible or maybe the classical hydrogenesis. These oxides, ferredoxin or other low redox electron carriers, both natural and artificial, in a readily reversible reaction. The hydrogen evaluation reaction in green algae is due to the reversible hydrogenesis. So, that means they are doing the back reactions over there. Then second one is called the membrane bound uptake hydrogenesis. These are able to take up hydrogen at low partial pressures, reducing a relatively high potential electron acceptor, but producing little or maybe no measurable hydrogen, means the hydrogen production quantity is very low in this particular case. Then the third one is called the nitrogenase enzymes. These normally reduces nitrogen to ammonia, but can also evolve hydrogen, particularly in the absence of nitrogen gas. So, reduction of nitrogen to ammonia, that is why it is called the nitrogenesis basically. Among the algae, only the blue green algae, which is known as the cyanobacteria, have these enzymes or maybe having this kind of particular properties. The presence of nitrogenesis and hydrogenesis have been found in photosystems and the fermentation bacteria respectively. Then this one is called the potential hydrogen producing microorganisms. So, in this particular case we have discussed detailed about the different types of microorganisms, then what are the scientific name of these microorganisms and what are the advantages and disadvantages. So, if we talk about the green algae, so basically the advantage is called the hydrogen production from water and high sun energy conversions. If we talk about the disadvantages also, it is having light requirement for hydrogen productions, because every time it requires the sunlight, and then sensitive to the oxygen presence. Then if we talk about the cyanobacteria, so this is the scientific name or maybe the microorganism names. So, it is also having certain advantages like hydrogen production from water by nitrogenase enzyme nitrogen fixation, and of course, there are certain disadvantages too. So, first it is called the inhibitory of oxygen for nitrogenesis, second one is the presence of oxygen and carbon dioxide in the product gas, and third is that sunlight requirement. If we talk about the photosynthetic bacteria, So, this is the scientific name of that particular microorganisms. It is also having certain advantages like can use different substrate for hydrogen production, and second one, use a wide range of light for hydrogen productions. Disadvantages: light requirement for hydrogen productions, water pollution problem by fermented broth, presence of carbon dioxide in product gas. And the fourth one is called the fermentative bacteria, that is Fermentobacter aerogens or maybe E. cloacae. So, these all are the scientific names. So, it is also having certain advantages, producing hydrogen without light demand, that is the biggest advantage over there. Second is can use a wide variety of substrates as carbon source. Of course, it is having certain disadvantages too, that water pollution problem by fermented broth and the presence of carbon dioxide in product gas. Now, we are going to discuss about the purifications of the hydrogen gas. So, the gases produced by biological process mostly contain the hydrogen 60 to 90 volume, and impurities like carbon dioxide, oxygen and small portions of moisture are present in the gas mixture. So, there are some kind of byproducts are already added with the hydrogen gas. Now, how we are going to get the 100% pure hydrogen gas? Scrubbers can be used to separate carbon dioxide. 50% W by V, weight by volume potassium hydroxide solutions is a good carbon dioxide absorbent. So, it can be used for carbon dioxide removal. Then alkaline pyrogallol solution can be used for the removal of oxygen from the gas mixture. Presence of moisture in the gas mixture must be reduced, otherwise the heating value of the fuel will be decreased. This can be achieved by passing the mixture through either a dryer or a chilling unit by condensing out of the vapor in the form of water, and then after that we are going to get the 100% pure hydrogen over there. Now, what are the criteria to choose the nanomaterials for biological hydrogen productions? So, it should be photoconductive, it should be high catalytic properties, it should be non-corrosive, and it should have high specific surface area. Now, what are the influence of the nanoparticles on biohydrogen productions? Till now we are discussing about different types of microorganisms, algae, bacteria kind of things. Now, how the nanoparticle will be able to produce the hydrogen gas or maybe help to produce the hydrogen gas in more volume concentrations or maybe in a lesser time, and efficiency of that particular system can be increased. The use of nanoparticles has been increasing significantly for applications such as protein immobilizations, biosensors, biofuels and microbial metabolic activity for hydrogen production. Thus, a positive effect of various NPs including silver, gold, copper, iron, nickel, palladium, silica, titanium, activated carbon, carbon nanotubes and composites were observed on BHP. These nanoparticles must be stimulating BHP by their surface and quantum size effect. Nanoparticles has larger specific surface area which enables strong ability to absorb the electrons. The extent of the quantum size is directly correlated with the rate of electron transfer between nanoparticles and enzyme molecules such as hydrogenases, which is known to catalyze the conversion of hydrogen to proton and vice versa. Now, what are the advantages? So, it is the effective waste management system, because we are taking all the waste products over there like algae, some kind of microorganisms, some kind of sunlight, some kind of other energy sources, prevents the environmental pollutions. Hydrogen can be produced with and without light in few methods. Of course, it is having certain disadvantages, need extra efforts to remove the impurities like oxygen, carbon dioxide and the moistures from the system, need more research for bringing this technology from lab to the industrial scale. That means, the commercialization of this particular technology needs more attention for the future hydrogen production point of view. Now, we have come to the last slide of this particular lecture. So, in summary we can say that in this particular lecture we have discussed about the biological hydrogen production process, which has been found to be more environment friendly and less energy intensive as compared to thermochemical and electrochemical process. In indirect bio photolysis, the cells take up carbon dioxide fast to produce cellular substances which are subsequently used for hydrogen production. Dark fermentation technology has the simpler reactor design and less energy requirement when compared to other hydrogen production technology. Hydrogen production efficiencies of MECs is up to 80 to 100% are significantly higher than that of the DA process, means dark fermentation process, of about 33% and water electrolysis process of about 65%. Thank you.